So, cytoskeleton remodeling and cell migration have an essential function to many biological processes. So, for example, homeostatic processes. Homeostasis is any self regulating process where biological systems tend to maintain stability while adjusting to conditions that are optimal for survival. If homeostasis is successful, life continues. If unsuccessful, disaster or death ensures. An example would be establishing an effective immune response or to repair damaged tissues. For cell repair, when a damage occurs for cellular repair to happen, the cells will send repair proteins to the damaged spot to quickly resolve it. In the process of repairing itself, it may be destroyed or converted to a cancer cell. For cell death, for example, cancer cells frequently respond to their altered state by undergoing programmed cell death and remain highly dependent on certain survival signals from their environment. So another aspect of this article is the assembly of actin stress. So it's regulated by a signal cascade when stress fibers function to counter membrane tension and to keep the non-adherent regions of a cell straight. The activity of the Rho GTPases is finally regulated by GTPase activating proteins, also guanine nucleotide exchange factors and guanine nucleotide dissociation inhibitors. So, actin-associated proteins can also block the degradation of Rho A and can lead to stress fiber formation. So, another important factor is cell migration. So, the proteins that are involved is the cyclin CDK inhibitors of the CIP and KIP family. So, cell migration occurs when a tissue or cell needs to be renewed or repaired. Older damaged cells are replaced by the migration of newly formed cells. This event is essential to maintain tissue integrity and homeostasis. A CDK, which is a cyclin-dependent kinase inhibitor, is any chemical that inhibits the function of CDKs. They are used to treat cancers by preventing proliferation of cancer cells. The CIP and KIP families are types of cyclin-dependent kinase inhibitors that are involved in regulating the cell cycle. These CDK inhibitors help regulate the Rho/ROCK/LMK/cofilin pathway by decreasing the actin stress fibers, which then causes the increase of migration and invasion and metastasis in different cell types. So this whole pathway also composes the P21-CIP1, P57-KIP1, and the P57-KIP2 genes, which regulate the Rho, ROCK, LMK, and cofilin pathway. So the Rho/ROCK signaling pathway is a major pathway transducing inhibitory signals, and it suppresses central nervous system regeneration in cells. It also regulates cell morphology, polarity, and cytoskeletal remodeling by regulating actin and cell migration, which is a very important aspect of this article. So there are different methods used throughout this study. So first being plasmids. So the plasmids that were used is called the pcDNA 3HAP57-KIP2, which is human, and pcDNA 3HAP57-DK-mut, which is the mouse, were employed. Also, the MYC LIMC1 and the MYC LIMC1 D460A mutant and MYC LIMC1 T508A are all present in the FPC1 vector. Flag LIMC1 and flag LIMC1 D46A mutations are present in the CMV vector, which was also employed within the GFP vector. Next, for cell culture and transfection, a human cervical carcinoma is known as HELA. So COS-7 is an African green kidney monkey. Human embryonic kidney cells HEK293 were grown. The cell line was created using HELA cells with tetracycline-controlled P57-KIP2 expression. The negative controls included HELA P57 and HELA CTRL to prevent the expression of P57-KIP2 and the TET on cell line. 2 grams per milliliter was added to the culture media for 24 hours. Lipofect amine plus for HELA and COS-7 was used for transfection. After transfection, the cells were harvested 24 hours later. So basically, transfection is a technique for genetically altering cells by introducing foreign nucleic acids into them. Transfection is a useful tool for investigating gene activity and control as well as protein function. Next, for immunoblotting, which is also known as Western blotting, so that's a sensitive method for detecting proteins such as like antigens or viruses and other plant diseases. This method employs a combination of gel electrophoresis and antigen antibody interaction. So, fourth, cell cycle distribution is a cell cycle's most basic job is to duplicate the enormous amount of DNA in the chromosomes and then divide the copies into two genetically identical daughter cells. So DNA flow cytometry was used to evaluate the distribution of cells in the G1S, G2/M cell cycle phases. So the cells were extracted and then fixed for 1 hour in cold 70% ethanol in the presence of RNase A. Samples were washed in phosphate buffered saline and stained with probidium iodine. So the last method, that is abbreviated as FRAP, is a microscopy-based method used to study the mobility of fluorescent molecules. FRAP helps with identifying ligand binding sites and understanding cellular transduction, which is a useful tool for the purpose of the study in order to visually see and analyze the data.